Good afternoon, everyone, and welcome to the Infection Prevention Strategy Tips Masterclass webinar. 2 weeks to 2 hours, rapid in-field detection of Legionella. My name is Dr. Rodney E. Rohde, and I will be the moderator for today. Despite great strides in investment in water modern treatment, practices designed to reduce waterborne pathogen outbreaks, Legionnaire's disease has become the leading cause of waterborne illness in the United States, and cases have been climbing steadily for decades. This is primarily due to the favorable conditions present in the built environment for amplification of Legionella bacteria, a diverse group of species that can cause disease when inhaled in contaminated aerosols. Understanding the current water management regulatory framework, as well as the gaps that exist, are key to our understanding where we are today and how we can chart a path to future improvements. Our learning objectives today are posted on the screen for your convenience. We'll be discussing Legionella and why it's considered a public health concern, how the COVID-19 pandemic has impacted cases of Legionnaire's disease, the pros and cons of traditional and new testing methods for this particular bug, Legionella, and how we can reduce the risk with a proactive monitoring plan. Participant slide lines have been muted. If you're looking for CE credits, we are offering 1 CE for nurses. You will receive an email after the webinar. Be sure and fill out that survey to receive a full 1 CE credit. By participating in this webinar, you acknowledge the statements and information provided are for general guidance purposes only. This webinar does not replace the guidance and direction of your local government and health authority. Participants should contact their local government and regional health authorities for specific instruction on all topics mentioned in this webinar. My name is Dr. Rodney E. Rohde, and I am a Regents Professor and University Distinguished Professor of the Clinical Laboratory Science Program in the College of Health Professions at Texas State University. I'm also the Associate Director of a university-level research center known as the Translational Health Research Center. I've been a member of the TIPS board for several years now, and it is my pleasure to be a part of this as your moderator. This presentation will last about 50 minutes, followed by 10 minutes for questions and answers. If you have any questions or comments, please type your questions in the webinar panel labeled Q&A, and we will follow up after the webinar to address any questions we are not able to answer during the webinar. The webinar series is hosted by the Infection Prevention Strategy, TIPS. TIPS is a not-for-profit that exists to advance innovations, ideas, and processes that make a difference in global health. TIPS extends to over 30 countries is a key strategic partner for many notable organizations, initiatives, and events. In the past few years, TIPS has contributed millions in scientific impact. Many of our successes are published on our open access journal, and you can find that at www.infectioncontrol.tips. Our motto: join, contribute, make a difference. Our sponsor for today's webinar is Lumen Ultra, and I'll turn this over to our speaker, Jeremy Duguay, to discuss the sponsor.
Thanks, Rodney. Just a little bit about Lumen Ultra before we get started. Lumen Ultra has been pioneering equipment and assay development to aid in microbiological monitoring for the better part of 2.5 decades now. We started out producing the world's only ATP test kits capable of measuring the microbial health of activated sludge in a wastewater treatment setting, and have since expanded that technology platform to allow for the detection of specific microbes through qPCR and next generation sequencing. Their head office is located in Fredericton, New Brunswick, Canada. Lumen Ultra now has a global presence with 6 offices worldwide that support nearly 4,000 clients across 80 different countries. Our commitment to excellence is verified through the company we keep, with a few key examples listed at the bottom of the slide here on screen. Most recently, through the COVID-19 pandemic, Lumen Ultra was able to answer the call from our Prime Minister for help securing COVID-19 testing reagents. Our CEO took immediate action to pivot our manufacturing business to become the primary supplier of COVID-19 test reagents for clinical tests, and that was to the Public Health Agency of Canada. And we were able to supply 500,000 test reagents per week during the first year of the pandemic. Lumen Ultra's product lines include fast, accurate, and portable microbial monitoring solutions that help customers understand microbial impacts on their process and provide peace of mind so we can all live, work, and play with confidence.
It's now my honor and privilege to introduce today's speaker, Jeremy Duguay. He is a molecular biologist with over 15 years of biotechnology experience in the public, private, and academic sector. With a wide-ranging background in technical sales, customer support, and research and development, he takes pride in understanding customer needs and helping customers solve those complex problems. An educator at heart, like myself, Jeremy is driven to help clients navigate and succeed in the constantly evolving world of microbiology. I will now turn over this webinar to Jeremy.
Thanks, Rodney, for the introduction, and I wanted to thank GBAC and TIPS as well for the opportunity to present today. Thank you to the audience for joining us and spending some time with us, and hopefully by the end of this webinar, you're going to have a little bit more appreciation and some knowledge regarding proactive Legionella monitoring and how proactive monitoring can help reduce risk of Legionella. Before we get into too far into the proactive monitoring piece, I did want to just provide a little bit of history first on Legionella. It was first documented as a potential pathogen in 1976, now in somewhat of a famous or infamous event of American Legionnaires conference in Philadelphia. There's about 2,000 attendees at this conference, and after that conference, there was several cases of illness that developed from the attendees. In particular, 182 clinical cases of disease occurred, and 29 individuals died after attendance at that conference. There was at the time, Legionella wasn't known as a pathogen, so there was a post-event investigation to try and determine what happened and what caused the illnesses and the deaths. And subsequently, after a CDC investigation, Legionella pneumophila was identified as the bacteria responsible and was so named as Legionella pneumophila at that time. So now, just a short 50 years later, there's more than 60 different Legionella species identified to date, and greater 50% of those are associated with some form of human disease. As well, Legionella is now responsible for the most water-associated disease outbreaks in the US. As you can see in the chart to your right, a significant number of outbreaks occur in the US. This is data reported from 2013 to 2014. Bacteria, specifically bacteria of the Legionella type, responsible for over 57% of those waterborne disease outbreaks. So Legionnaire's disease itself falls under a group of conditions that are referred to as legionellosis. Legionellosis encompasses both Legionnaire's disease, which is the more severe pneumonia that is fairly fatal in people that acquire it. And Pontiac fever is a milder form of the pneumonia that most people resolve on their own without any intervention required. So we hear more about Legionnaire's disease because of its more prominent fatality rate. There's about 485,000 annual cases of pneumonia per year in the US. Roughly 15% of those are linked to Legionnaire's disease. So again, quite a few cases per year. And of those cases, the 15%, 90% are shown to be due to the Legionella pneumophila species. However, there is some debate in the literature about whether that is truly that high, that percentage, and whether other species can also contribute significantly to disease and mortality, because of the specific diagnostic tests that are used are kind of skewed towards Legionella pneumophila. There's, the disease itself carries about a 10 to 30% mortality rate when infected, and typically those with immunocompromised systems, the elderly, or those with pre-existing conditions, particularly of the lung, or unique situations where people have smoked and are at higher risk. Those are the cases where the mortality rate tends to increase closer to that 30%. The general population, it would be a little bit lower, obviously, than that. It has a significant contribution to the healthcare system, financial cost and impact of greater than $600 million per year, US. And the disease itself is acquired by inhaling or aspirating contaminated aerosols or micro droplets. So the contaminated water has to be inhaled into the lung or aspirated if drinking down into the lung as well. Legionnaire's disease itself has been increasing quite a bit over the last 18 years. We can see on our graph here 6 times from 2000 to 2018. Annual cases went from just about 1,000 to over 10,000 on the graph in 2018. Reasons for this are thought to be many and potentially include the aging population, as well as an increased population density. So people are moving into more urban areas and settings and higher density areas. Also potentially green building initiatives where things like low flow water and decreased water pressures have caused more water stagnation, which is a ripe condition for Legionella to proliferate. There's some speculation about climate change and global warming also contributing to this increase over time, as well as a focus on enteric disease, water pathogen treatment and eradication, as opposed to Legionella, which is more of a respiratory illness. And finally, there is also some speculation that improved diagnostic testing can be contributing to the increase as well. But we also know that it's probably a vastly underreported disease because most diagnostic tests don't confirm in pneumonias what the source of the bacteria was that caused it. So a lot of potential reasons for the increases, but it's of note that there's been significant increase of Legionnaire's disease over the last 18, 20 years. Legionella in the natural environment are present in low levels naturally. They're not in high levels just in natural bodies of water. They're also found in some soil environments. But they do tend to thrive in built environments, and particularly in protozoan hosts that thrive in biofilms under warm water conditions. And the built environment does lend itself to that type of condition. So you tend to see an amplification of Legionella bacteria in those built environments. The favorable conditions for proliferation include a temperature range of 20 to 45 degrees Celsius. That's the favorable temperature range for Legionella. When it's less than 20 degrees Celsius, the bacteria become dormant. And when it's greater than 60 degrees Celsius, the bacteria will die. So there are mitigation efforts to control bacteria in warm water, hot water tanks or lines by setting the temperature greater than 60 degrees Celsius. Of course, you just have to take care at that point to make sure anybody that's using that water is not subjected to any harm from that hot or scalding water. As mentioned, water stagnation, when water sits, it provides the right condition for bacteria to attach to surfaces and come into contact with those surfaces and form biofilms. And when those biofilms form, again, the bacteria tends to thrive in the biofilm environment where there are protozoan hosts. So the built environment provides kind of the ripe, perfect conditions for biofilm formation and for Legionella proliferation. So anywhere there's pipes feeding water into a system, building, or a process, there is the potential for biofilm to form. And when biofilm forms, Legionella tend to do quite well because they are able to proliferate within that biofilm, as well as infect and invade the hosts that they prefer in those biofilms as well. They also exist in the free-floating water, but we'll talk a little bit more about that in the next slide. But all of these examples on the screen are areas where aerosols might be generated after Legionella have inhabited and amplified in the water system. Things like decorative water fountains, cooling towers, faucets, hot tub saunas, all potential sources for Legionella amplification and aerosolization and potential infection. So the Legionella life cycle looks a little bit like it's, as mentioned, the bacteria likes to thrive in biofilm. And when it's in the biofilm, it has lots of nutrients that it requires for growth, and also host organisms where it invades the host, typically amoeba. And once it's inside the amoeba, it will rapidly replicate. Once the nutrients within the amoeba are exhausted, the Legionella will break out of that host and then look for new hosts. And that will be generally, in the water column, planktonically, so free living, where they'll look for more hosts within the biofilm if they have the opportunity to, or if they also are aerosolized and are able to find a human host, they will then enter the lung if possible, and they can infect the alveolar lung and cause disease. So the life cycle is kind of repetitive in that sense. They'll replicate within the host amoebas if they can within the biofilm, enter into the water column, and then repeat that cycle until they potentially are aerosolized and exposed to humans. So this really kind of is highlighting the importance of controlling Legionella in the built environment, and really looks at some of the unique risks that are presented to the built environment. And uniquely, we're going to talk a little bit about premise plumbing as it presents and represents some unique risks. And when we're talking about premise plumbing, we're talking about once water from a municipality, a main line, is distributed to a building, it's kind of referred to as premise plumbing at that point. That building has its own unique water use and plumbing concerns, and it could include anything like multi-unit residential buildings, public buildings, hospitals, long-term care homes, schools, hotels, etc. And there's a lot of water sources and water uses within those buildings, including everything that we've talked about previously where Legionella can become problematic. Things like cooling towers or decorative fountains, ice machines, sinks, showers, faucets, hot water storage tanks. So all of those risks within a building need to be kind of identified and monitored for reduction of risk in that unique premise plumbing environment. So that kind of brings us to some of the unique possibilities due to or some of the unique concerns associated with COVID-19 and the lockdowns that occurred over the last 18 to 20 months, and how that can increase risk for Legionella. So there was a publication here in September of 2021 in the American Journal of Infection and Control, where they looked at the increased risk of Legionella in buildings that were kind of shut down or had reduced occupancy or reduced access during the COVID-19 pandemic, and really found that, just as was speculated, that when water was sitting in buildings and not moving through, stagnant, and had less occupancy, there was much greater risk of microbial contamination, in particular by Legionella in some of these building water systems. And that, again, that was linked to that extreme water stagnation, to those closures. So this, obviously was speculated and was, but confirmed through some testing, but also underlines the increased need for vigilance when reopening buildings after being shut down or reduced occupancy over time. So because these prolonged shutdowns have provided favorable growth conditions for microorganisms, including Legionella, there's a lot of reasons for that. Disinfectant residuals are not there. That water stagnation, warm water conditions due to the water sitting. There is a need for strong reopening plans for these sites, particularly to reduce the strong extreme risk that occurs when Legionella proliferates in those stagnant areas. So there are controls that can be implemented. There's lots of things you can do, and there are sources of information where you can go to get standard practices to reopen a building after COVID-19. And a lot of those things include, flushing the lines, increasing temperature in hot water systems, cleaning different areas that are known trouble spots, and of course, testing to confirm as well. So several organizations have, offered their advice and expertise to help with water management programs, and that includes for reopening buildings after shutdowns due to COVID-19. Some of those include the Association for Water Technologies, the American Society of Heating, Refrigerating, and Air Conditioning Engineers, or ASHRAE, European Center for Disease Prevention and Control, same Center for Disease Control in the US, and the UK Health and Safety Executive. They all have guidelines on how to properly monitor a building's water system for Legionella risk, and how to mitigate the risk associated with that bacteria. One of the key themes and elements throughout all of these different programs is that testing is a key pillar to the water management program. So in order to inform how effective your treatments are, how to inform how effective your mitigation efforts have been to eliminate or reduce Legionella risk, you actively have to test to be able to confirm. So that's kind of moving us into the next section of the webinar, where we want to take a closer look at the testing landscape for Legionella risk management. So traditionally, microbial monitoring for Legionella has been done through culture-based testing, particularly for environmental monitoring or routine monitoring of a building's water system or any water system. For Legionella risk, we have relied on the industry standard culture method. It's been used for about 150 years. Just a brief refresher, it requires nutrients and specialized growth conditions to enable bacteria present in a sample to grow to visible colonies, where we can then count them to determine how many are present in the sample. It generally requires the use of aerobic plate counts or heterotrophic plate counts to do so. And there are some portable methods as well that are available, things like dip slides as present on the screen, which enable you to take that out of the lab and into the field. So again, very effective methods that have been used for quite some time. And there are some limitations that are quite well known about some of these methods as well. So the main factor is that it takes time to get results through culture. So particularly for general microbial indicators like HPCs or general microbial contamination, it takes 24 to 48 hours to get results. They can be inaccurate in that they don't quantify the total microbial load that's present in a system. So heterotrophic plate counts only count organisms that consume carbon in their growth process, and aerobic organisms at that. So they tend to report about 0.1 to 1% of the total microbial population that might be present in a system, and certain microbes cannot be cultured at all. And that includes Legionella. They don't grow on the standard heterotrophic plates. The analysis of the plates can be subjective, so it requires some human interpretation. Colonies can vary in size, thickness, and counting them by eye sometimes can be laborious and difficult to do. You sometimes get clumping of cells and colonies which grow together, which leads to some large degrees of error. And as mentioned, it's not specific for Legionella risk. So there are specific Legionella-specific culture methods as well that have been developed over time to kind of be able to mitigate and identify Legionella-specific risk, and that's the buffered yeast charcoal extract plate method, which is the predominant standard method that's used. It provides unique nutrients that Legionella require to grow on the plate. It is, again, highly specific for Legionella, but it comes with some challenges. It takes a very long time to get results with this method, anywhere from a few days if there's a lot of bacteria, typically around 7 to 14 days for results. It doesn't capture the viable but not culturable fractions, so cells that are still alive but not able to be cultured won't appear in the plates. It can be prone to interference from other cohabitant microbes, so those need to be typically pre-treated to remove that interference. It is specific for Legionella pneumophila, but it also does require specialized laboratories to get your results, and that usually requires shipment of at least 1 liter of sample with undercooled conditions to get it to the lab in a short period of time for testing. So again, very good tools, but they have some severe limitations. And if we kind of look at the overall strategy of using traditional culture tests to identify, proactively identify risk in a system, whether it's a building or a water system of any sort, we have to kind of follow the strategy of collecting and shipping samples to the lab, and that can typically take anywhere from 7 to 14 days to see what kind of Legionella control we have in our water system. And then if we identify a problem, we would take a day or two to return to the site and take some mitigation effort to reduce that risk from Legionella. And then we want to assess how effective that treatment or mitigation was. And then we have to retake our samples and send them back to the lab. Again, wait for another 7 to 14 days for our results to come back to see how effective our overall treatment was. And so that results in a very long cycle time of 15 to upwards of 28 days for results to be able to identify a problem and then confirm it's been treated properly. So as a routine kind of condition monitoring tool, just the culture test has that severe limitation. If you have to wait that long to know you've solved the problem, there's a lot of risk associated with what's happening in those previous 28 days. So you can either take the overcautious approach and shut a building down, or over treat it, but neither of those are good options for really mitigating the risk to occupants. So this has kind of led to an evolution in the Legionella testing landscape. As mentioned previously, cases are increasing. More and more outbreaks of waterborne illness are being detected and impacting people. So there's a need to be more proactive in reducing and mitigating the risk of Legionella. And that's really been driven by economic and technological improvements. The culture methods still exist and are still very valuable for regulatory-based purposes and also for confirmatory purposes. But we're really kind of seeing this enhanced rapid methods which are coming on the market, which are now kind of allowing us to be much more proactive in our Legionella risk mitigation. So I want to talk a little bit about some of those rapid methods for Legionella detection now, and what they involve and how effective they are. So there's quite a few different types of rapid methods that are out there, and I'm going to kind of highlight a couple of them. So there's things like the liquid-based most probable number, so a culture-based method that's a little bit easier and faster to get results compared to traditional culture. There's the direct fluorescent antibody test, which labels the antigens with antibodies for visualization and detection. Lateral flow antigen tests, which are simple to use, but sometimes have some specificity and sensitivity issues, as well as qPCR. And that's the one I'm going to focus on for the majority of today's discussion. And it's really been the method that has kind of taken the lead in a lot of environmental and industrial applications for monitoring pathogens in those applications. And it really, qPCR has come to the forefront over the last 2 years as well, as it was, the primary method for detection of COVID-19 in clinical assays, as well as in environmental assays for mitigating risk of COVID-19. So ANSI ASHRAE Standard 188 Guideline 12-2020 does list the use of qPCR for negative screening of samples, and it's an excellent tool for this. Most standards still cite the culture method as required, particularly for outbreak investigations, and that's again primarily due to its long history and use, where action limits and thresholds have been developed around how much Legionella is too much and what you need to do to mitigate risk in a system. And what actions to take when you have certain levels of Legionella in your system. qPCR is not quite there yet from its threshold levels and action levels, but they're very quickly being developed and reproduced and published in literature sources for people to use as a starting point for taking action based on qPCR as well. So want to talk a little bit of the basics of qPCR as well, because I know some people are familiar with it, obviously, after the last 2 years where it's been quite heavily used and talked about in COVID-19 clinical detection, as well as environmental detection, but not everybody is super familiar with the technology behind it. So it is a technique that enables the monitoring through amplification of target DNA of an organism. It's fully quantitative. It is the gold standard method in applications like molecular diagnostics, like, for example, COVID-19. But it hasn't really gained that same acceptance in a lot of environmental and industrial applications, but it is kind of getting there rapidly. So that's where we're going to kind of focus our discussion about it today. So just to give you a little bit about how it actually works. So, all organisms contain DNA as a genetic blueprint or code for genes that eventually code for proteins in a system. And these proteins eventually form some function or perform some function within a cell. They're based on those DNA sequences that encode the genes. And those DNA sequences are made up of just nucleotide bases. There's 4 of them, A, G, C, and T. And when they run into thousands of sequences, you end up with a gene, and that gene sequence is usually unique for an organism or for a particular function of that gene. So Legionella, if we're talking about specifically about Legionella pneumophila monitoring, we can determine a specific gene sequence that only Legionella pneumophila has, for example, the MIP gene. We can also target a specific gene sequence for, say, Legionella pneumophila serogroup 1 through the WZM gene. Or we can talk more broadly about the species or all species of Legionella through targeting a region of the genome that is highly conserved and doesn't differ much between the different Legionella species. And that would be through something like a 23S ribosomal RNA gene. So again, uniquely conserved through evolution. And if you target this particular location of the DNA sequence, you're going to get all species of Legionella represented in your assay. So the name itself is qPCR, so it's quantitative polymerase chain reaction. We know that it's a quantitative method. So how does that work? Well, when we have our starting template and target, in this case, let's say it's Legionella pneumophila, from our sample, we would extract the DNA from that sample. And then if we're starting with the target sequence, and we would add primers and polymerase to amplify and copy that DNA. Primers target that specific sequence, bind to individual strands of DNA once it's separated through a heating process. This is all done in a qPCR machine. A thermocycler takes a couple of hours to do. You add the components into a tube and set it to go for those 2 hours, and maybe over 35 to 40 cycles of amplification, you'll eventually get a visual or visible amount of copies of your target. So the first cycle, you end up getting two copies of your target sequence. The next cycle, which takes maybe another 30-45 seconds, you get 4 copies. 4 copies become 8 copies become 16, and so on and so forth, until you have that 40 cycle sequence completed, and you have millions of copies of your potential target. And the way it's quantified is through the incorporation of fluorescence or fluorophores into the amplified DNA. So our primers or probes bind to that specific sequence of DNA and amplify through that process, and we have the copies made with incorporated fluorescence that then gets measured in a qPCR device, which measures fluorescence. Increased fluorescence leads to an increased signal. We can then quantify the DNA through standard curves, and the higher the amount of target, the lower we'll get an amplification event in our qPCR device. The lower the amount of target, the later in those cycles we'll see an amplification event where we get visible or detectable target. So a low CT value is a high amount of target at the start. A high CT value is a low amount of target at the start. If we don't get any amplification, that means our target wasn't present. We can compare our starting sample concentration back to the standard curve to determine exactly how much sample, how much target we had in our sample that we started with. A typical qPCR workflow will look something like this. So you'll start with collecting a sample. Typically, it could be a water sample, it could be a biofilm sample. You're going to concentrate and then wash your sample and then extract DNA. And you're going to lyse cells at that point to extract their DNA. You're then going to purify the DNA and then move it into the qPCR assay component portion of the assay, where the instrument will read fluorescence, as I mentioned in the previous slide. So you have lab-based as well as field-based methods that are available to do this. Typically, qPCR is a lab-based method, but in general, it has been also adapted to work out in the field by some manufacturers. There are advantages to qPCR compared to traditional culture-based monitoring. It is rapid, sensitive, accurate, and specific. There are also challenges with the technology. And so one of the challenges is the ability to differentiate between living and dead cells, because both can contain DNA. So as well as culturable versus non-culturable organisms. Interpretation, as mentioned, is also an area of a challenge as well, because we haven't the experience that the culture method has to get thresholds for all scenarios and circumstances. So that's being developed. And so qPCR can typically be used in several different areas of your Legionella monitoring. It can be used for outbreak investigations to get quicker results, maintenance verification, research, routine monitoring, certainly, as well as negative screening, as mentioned. So I did want to tackle some of the challenges with qPCR as well, to highlight what can be done to try and overcome some of these challenges. And the first challenge we see is a lack of correlation with colony forming units, the culture method that's kind of been used again for the last 50 years. It is true that CFU per mil or CFU per liter does not equate to the same thing as gene unit per liter. So they're two totally different measurements. So a culture test and a qPCR test. So we don't always get good correlation between the results. However, they are attempting to quantify the same thing. So 1 CFU per mil is 1 cell per mil. 1 gene unit per mil is attempting to quantify 1 genomic unit per Legionella per mil. There's good reasons why the results don't always trend in the same magnitude, and that's typically because, again, qPCR identifies things like viable but not culturable, as well as potentially dead cell DNA that might exist in a sample. So it tends to give you data that's about 1 to 2 log higher than culture data, and that's been published in the literature in several studies. But the results do tend to typically trend in the same direction. There are, again, scenarios where CFUs and gene units qPCR data do kind of trend similarly, and that's generally speaking when the microorganisms are growing in a growth phase where there's lots of nutrients in active growth. You do tend to see fairly good correlations between the qPCR data and the culture data. Under low nutrient conditions, you don't see as good of a correlation, and also under systems that are under good control, where there's biocide being applied or treatment being applied, and a lot of stressed or viable but not culturable organisms, again, we don't tend to see a great correlation at that low end of detection. And again, this is a reason why negative screening is recommended by ASHRAE for qPCR, because when qPCR gives a negative result, it is truly a negative result. If it's positive, there's you want to make a confirmatory test with a culture test to confirm that. qPCR challenge number 2, the live dead cell determination. There is some manufacturers that are using viability staining to try and overcome that. That's basically where the incorporation of a dye gets incorporated into DNAs to make it unavailable or not available for amplification. So the requirement is that a cell needs to have a disturbed membrane in order for that dye to be uptaken into the cell and then intercalated between the DNA fragments so that they're not available for amplification in the qPCR event. So only the theory goes that only living cells with intact membranes can then be detected by the qPCR. The problem is there's limitations with that because certain methods, certain organisms, certain treatment types will impact the reliability of the assay. So it's been described as more of a qualitative measurement rather than a strictly quantitative at this point in time. As well, there are some requirements for lab-based instruments to get the dyes, to use the dyes. So it's restricted to lab use. It is a promising method for future development, though, as we get more reliable data for different microorganisms and different treatment conditions and how they impact the dyes and their uptake by living cells or membrane compromised cells. Other manufacturers utilize filtration-based technologies to try to isolate the living fraction of bacteria that might be present in a water sample, for example. So they pass the sample through a filter and capture intact living cells. Any extraneous or extracellular DNA that's from dead cells passes through the filter. And if there's potentially viable but not culturable cells, they're also captured in this filtration as well. It doesn't work for all treatment methods, though. However, there's some types of treatment that might inactivate the organisms from replicating further, things like UV treatment. And so they're not able to replicate, but they're effectively dead but still have membranes intact. So they will be over quantified in that sense. So it is important really always to develop a baseline. And if you do have increasing levels of Legionella in your sampling over time, that can be attributed to growth from living cells, not from dead cells, obviously. And there is a recent example of this in the literature. So from February of 2021 in Microorganism, the journal Microorganisms, the first real longitudinal study looking at qPCR compared to culture methods in cooling towers that were experiencing in an industrial complex kind of severe Legionella outbreaks over the years. So this group instituted a comparison of qPCR testing for Legionella species in comparison to culture testing to see if they could get a better proactive measurement and of risk of Legionella in that system. And what they did find after this 39-month trial, where they took hundreds of measurements with qPCR as well as culture method, the culture method wasn't picking up any Legionella in their system, where the qPCR was detecting Legionella species quite significantly. They were able to develop a threshold baseline for their cooling tower system. And then once levels were above that threshold, took action in their system to investigate why the levels were above that threshold, and then were able to mitigate and reduce risk from Legionella, and actually found qPCR to be the better tool for that purpose. And in their testing, actually culture, they stopped testing for the most part with culture and just relied primarily on the qPCR where appropriate to do routine monitoring and identify risk in their system. So just wanted to give a bit of a brief summary of the methods that we kind of talked about. So there is specific culture method, the qPCR method. There are other rapid methods like intracellular ATP as well, and heterotrophic plate count as a general indicator of microbial risk. So we go from kind of the more specific methods at the top, specific culture and qPCR, down to the less specific at the bottom, the second gen ATP as well as HPC or general culture. So qPCR advantage is very rapid, proactive, gives results in about 2 hours. The culture test very specific for Legionella risk, but also takes quite a bit of time. And then the other tools are very good at general microbial indications, but again, they don't give that specific risk overall for Legionella. So just want to talk briefly about some applications of qPCR to reduce Legionella risk, and kind of highlight an example of where it can be used in a hospital environment, for example. If we had a hospital that was receiving municipal water, what we would want to do is look and map out the overall flow of the water within buildings and identify all of the water related risks that might be present in that building, and then identify critical control points within those high possible risk points for identification for microbial monitoring. We're going to collect samples from those critical control points and then test them for Legionella using qPCR within 2 hours to get results. And then we're going to determine where the problem areas are and continually monitor over time. And as we do that, we're building our baseline to set our thresholds for action. So in a hospital environment, it might look something like taking a couple of points where we've identified increased risk. Maybe it's a hot water tank. Maybe it's a specific wing of the hospital that has low flow zones. And we're going to continually monitor that with qPCR on a defined basis. And then if we see any sample events where we have high elevated qPCR data relative for Legionella, we're going to take some action, apply a treatment, and then very quickly identify how effective that treatment was. And also this allows us to go out into the system and identify if there's some reason for that rapid influx in Legionella. So perhaps a valve gets shut off and water's not flowing in a particular wing of the hospital or something like that has caused water to stagnate and cause higher than normal values of Legionella to occur. So it gives us that ability to investigate challenges that occur and really mitigate against them from happening in the future. So it really brings us to the really the kind of summary slide here, and it's really the proactive difference for monitoring with qPCR compared to culture test. Now we can go out into the field, collect samples, and test them on site within field qPCR testing, get our results in just a matter of a couple of hours. If we identify a problem, we're going to enact some mitigation. It could be a couple hours, it might be a day or two, but we then can reassess how effective our treatment was, again, within just another couple of hours. To monitor how our control has worked to reduce Legionella. So now we taken that cycle time that was previously with culture testing potentially 10 to 28 days up to or down to less than 1 day, where you can proactively find problems and solve them on the spot. So that kind of brings me to the end of the formal presentation that I had for today, but happy at this point to address questions that folks had throughout the presentation. Okay, so I'm gonna go to some of our questions that I see here that have been logged throughout the presentation, and I can tackle a few of those. So how long does the temperature require to be at 60 degrees Celsius to kill the Legionella is one question we have here. Legionella, when the temperature range is between 45 to 60. That's a really good question. It does take some time at 60 degrees Celsius for the Legionella to be completely killed. I'm not 100% certain of the exact amount of time, but it would require at least an hour, I would think, at that temperature to ensure complete eradication. So you want to make sure that it's at that 60 degrees Celsius for quite some time. In the lower range, temperature range, 45 to 60, you might get some death of the Legionella, but not complete eradication. So that's where you want to make sure you're hitting that 60 degrees Celsius temperature for quite a while. There's another question here. Is there a correlation between qPCR and standard culture tests for threshold action levels? And it's a typical question that we tend to see. And in certain circumstances, you can actually correlate qPCR with the standard culture method. The problem is that it tends to be unique to each process and application. So if it's a potable water situation with a certain biocide being applied, or whether it's bleach or chlorine or monochloramine, the impact on the Legionella and how that's detected by culture, as well as the impact and how that's detected by qPCR, will be fairly straightforward. So you can develop a correlation in those unique environments and set some thresholds for your unique application to be able to actually make that correlation for your system. the challenge is getting large data sets, I guess, over time for unique situations and applications. Things like cooling water applications, things like potable water, drinking water applications, in getting large data sets of comparison for, with all parameters controlled for, to be able to get those thresholds kind of generalized for each application. That's going to take some time. So that's going to, be, it took, 100 years of cultures to get really good, correlations and thresholds for some diseases and applications. So it's going to take a little bit of time for qPCR to get there, but the data is evolving quickly, and people are coming out with more and more information about it every day. So I would expect it's just a matter of a few years before we start to have those standard correlations between culture and qPCR threshold action levels.
Great answer, Jeremy. As a clinical microbiologist, I can certainly appreciate these kind of nuances with these really difficult to culture organisms. One of the questions that has popped up and I think is on the mind of many people is, what exactly is VBNC, that viable but not culturable? And can you elaborate just a bit more on that? I think the audience, not being all of them being clinical microbiologists, might want to hear that a piece of information.
Yeah, sure. I can elaborate on that. So viable but not culturable organisms are, as the name states, they're microbes that don't show up through culture method. So they exist in nature and in the environment, in your various water samples and soil samples. But if you were culturing for them, you would never know that they existed, because they don't actually aren't able to be cultured. So with the advent of some of our new molecular tools, things like qPCR, things like next-gen sequencing, we've been able to get a better understanding that these organisms actually exist in a state where they're essentially, for the most part, they're viable and living, but they're just not able to be cultured. Whether sometimes that's because there's unique growth conditions that are associated with that particular microorganism, so they might need an extreme temperature or an extreme nutrient that's not typically available, or just the fact that they're stressed and kind of entered into a state of dormancy, for lack of a better word, where they're preserving their energies and making it so that they're just resistant to active growth and replication, and waiting until the proper environmental conditions occur for them to do that. So the state itself is, it encompasses many different things for many different microorganisms, but essentially what it comes down to is that those organisms will not be cultured by any method, and we need to rely on other tools to be able to identify them. And the advent of molecular tools has really kind of helped spur their acknowledgement and existence, in the last 10 to 15 years.
Yeah, I totally agree. As someone who sits at the intersection of clinical microbiology and molecular diagnostics, I can certainly appreciate that answer. I know another question here that you might want to address, because everybody in the world right now is hearing about false positives and false negatives and things like that. Can you talk about any concern with respect to the qPCR method, specifically around false or negative types of issues?
Yeah, certainly. So it is definitely top of mind with the COVID-19 discussions about this type of thing. qPCR is highly specific, so it tends to be, obviously it's dependent on your assay that you've developed and the targets that you're using to amplify the DNA. But if you choose your targets appropriately and you validate your technology appropriately, you can determine a very specific region of DNA that only one organism contains, specifically like Legionella pneumophila, for example. And then as long as you validate your assay appropriately, the chances of having a false positive with qPCR are very low. So, greater than 99.9% accuracy with a highly specific assay. Now, so, if you're looking to validate an assay, there is an ISO standard, for example, for qPCR technologies looking at Legionella in particular, and they recommend one of the validations is to be, to be, if it's a species assay, it has to be able to detect, 40 different species of Legionella, and not detect 140 different cohabitating Legionella organisms that cohabitate with Legionella. So microorganisms that you would typically find in the environment with Legionella are not positively identified by your assay. So that's a requirement by the ISO standard. So that's a really good way to make sure you're not getting false positives. And then on the false negative side, again, the qPCR can pick up on dead cell DNA sometimes if that type of DNA is captured in your assay. It's not necessarily a false positive in that case. It's just really detecting DNA that's been expelled or excreted from a lysed cell that maybe hasn't degraded yet in the environment. So it is kind of an overrepresentation of the active growing risk in your system. But if there was Legionella, for example, in your system, and it recently died and there's some dead cell DNA, that means that Legionella is in your system somewhere and needs to be, continually monitored to make sure that it doesn't increase to cause problems. So knowing about its existence, even if it's dead, can sometimes be beneficial to have that information that it's around.
All right. Yeah, great answer. That's really important information. I've got a couple of questions here I thought you might want to address. Perhaps it's on the side of sampling. Do you have any experience, or can you talk a little bit about not only kind of where to sample, but also maybe how that is properly submitted for Legionella testing with qPCR?
Yeah, it's again a really an important part of the qPCR process in any testing program is to make sure, and it's something I think that often does get overlooked quite a bit, not just for Legionella, but for a lot of microorganism detection and testing. Is this is sampling a good sample? Everything starts with a good sample, so a good test result always starts with good sampling. There are certain guidelines that the CDC puts out, as well as ISO and ASTM. All the regular standard providers have methods and that you can follow to kind of generally collect a good sample for Legionella. In general, depending on the type of water that you're testing, there may be some specific test sampling protocols that you want to follow. So if you're testing potable water versus non-potable water, there'll be some unique characteristics and things to follow for the sampling, particularly for non-potable water, things like cooling towers where there's a risk of aerosolization. You want to be really careful about turning off those aerosol generating fans and things before you go in and collect samples to mitigate your risk. But yeah, in general, you want to collect representative samples from the process, and depending on what type of investigation you're doing, if it's a routine monitoring audit where you're going in and looking for Legionella to identify your critical control points, you want to cast a wide net, but you also want to hone in on spots that you suspect you're going to have, potential problems. If you're looking at a hospital environment, for example, with shower heads that have some, growth layers on the shower head, and some buildup that's occurring there, you might want to target those for a biofilm swab to be able to measure if that buildup is actually biological and if there's Legionella specifically within it. Other cases, if you're collecting water from a tap in a hospital system, you want to see if there's Legionella in it, you might want to take that first flush sample to be able to tell if there is Legionella in the premise line. If you flush it for a couple of minutes before you collect your sample, and you're ending up with water that's coming from the municipal feed after you flushed out the premise line, you might end up with really low or really good control in your system or in that test. But if you haven't tested that first flush where it's sitting in the premise plumbing in the pipes, then you're kind of missing that risk from that premise, that location. So yeah, there's a lot of different guidelines out there that can help kind of to ensure that. But then beyond that, once you collect your sample, you really want to ensure that it's not changing over time. So you want to test it as quickly as possible. So if you're sending it to a lab for culture testing, send it quickly, 24 hours or overnight. And if it's going to be subjected to extreme temperatures, you want to cool it. So put it in a cooler with cold shipping. Otherwise, that extreme temperature could impact the microbes that you've collected. So there are quite a few best practices for collecting samples, and just a couple of them that we talked about here, but certainly, you can seek more guidance from some of the major standards and guidelines out there.
Yeah, great reminders, Jeremy. I know, again, in my experience in both public health, microbiology labs, and the medical laboratory, one of my all-time credos for my students and colleagues is, you cannot put too much emphasis on the quality of the specimen or the sample, right? I mean, if we don't get the right specimen or sample, it really doesn't matter what type of detection system you have. You really need that sample to be in great shape. So I think that's a great question. How about one more? We're getting over 1 o'clock here, but there's one here that I thought might be kind of an interesting question for you. Do you have any examples, or perhaps just from your own insight of talking to customers, kind of their maybe testimonies about that system, that qPCR technology in the field?
Yeah, certainly. So we have some customers using our qPCR technology in the field as well, and certainly customers that are actively using the technology to try and prevent and be proactive in the Legionella monitoring. And we can, if you're interested in maybe hearing about those examples, you can kind of reach out to me, myself, or somebody else at Lumen Ultra, and we can walk you through that.
Yeah.
But one kind of recent example that I'll mention is we just recently submitted our qPCR testing to a CDC Elite lab for proficiency testing in comparison with their proficiency testing on the CDC Elite proficiency testing program. So basically, they applied our qPCR technology alongside their culture testing on those blind samples that they received as part of their proficiency testing. And we just got the results back, and it matched perfectly with the culture test in that case. Again, these are active, happy, well-adjusted microbes that are being sent to them for testing. And in our case, with our qPCR species testing and with our Legionella Pneumophila test, we were able to actively or accurately identify which of those blind samples contained Legionella species, and as well as which ones contained Legionella Pneumophila. So that's something to look forward to. We'll be promoting that a little bit more as we digest that report and put that out there into the world. All
right, outstanding, and congratulations on that. All right, thank you, Jeremy, for this excellent and very informative presentation. I also want to thank the audience for attending today. We certainly know everybody's time is valuable, so we really appreciate you hanging in there for some of the questions. Be sure and stay tuned. TIPS will be certainly sponsoring additional relevant webinars, so stay tuned, and we hope you all have a great day and a great weekend. Thanks again.
Thank you.